Hello my dear students, welcome back. How are you all? I hope all of you are fine. So in the first part of the video, means we can say the first part of this chapter, I discussed about introduction of the cell cycle and cell division. I discussed how many types of cell divisions are there. I discussed three types of cell divisions are there. Amitosis, mitosis and meiosis. Then I discussed the definition of cell cycle. The cell cycle I told you, the sequence of events that will take place in the formation of two daughter cells from one parent cell is called as what? Cell cycle. Then I discussed about the generation time. Generation time means the duration of the cell cycle. And the example I took, that is standard example that is taken, is of human cheek cell, that is the cell cycle duration is the 24 hour. Then apart from that, yeast also you should know, that has the generation time of 90 minutes. Bacteria have generation time of 20 minutes. Some examples you should know. Clear? Then in the last I discussed about the different events of the cell cycle. Means a summary I told you that cell cycle will complete in 3 steps: interphase, M-phase and the cytokinesis. Clear? Now today in this video I am going to discuss about the interphase. Actually, from where this word has came, interphase. This interphase word has came from the intermediate phase. Why it is called as intermediate phase? Because it is the phase between the two consecutive cell cycle. Means we can say the gap between the cell cycle. Suppose one cell has divided to form two daughter cells. Clear? Then again when that two cells are formed, when they will again undergo for the division, they will take some time. So the duration between the two consecutive cell cycle is nothing but the interphase. Okay. Then I discussed in the interphase maximum time of the cell cycle, the cell remains in the interphase. Means interphase may accounts for 75% to 90% of the duration of the cell cycle. For example, that human cheek cell I told you, that even though it have a generation time of 24 hours, but the division activity will last only for 1 hour. Otherwise, for remaining 23 hours, the cell will remain in the interphase. Clear? So this interphase is further divided into 3 subphases: G1 phase, S phase and G2 phase. So 3 phases are there: G1 phase, S phase and G2 phase. Now we have to study about them one by one in the detail. Okay, so that we will start one by one. So first one is the G1 phase. Some other names are there. G1 phase, or it is also called as post-mitotic phase. Post-mitotic phase, or it is also called as first gap phase. First gap phase. So different names are there: G1 phase, post-mitotic phase, or first gap phase, or gap 1 phase also you can say. In different books, different names are given. So you should know why it is called as post-mitotic phase. You see, when a cell has divided to form two daughter cells. So immediately after the cytoplasm division, or we can say immediately after the cytokinesis, the cell will undergo into the phase, and that phase that will undergo into the interphase, and that first phase of the interphase is nothing but the G1 phase. Okay, that's why it is called as post-mitotic phase, because it is the post means after, it is the phase just after the cell division. Clear? Now something I want to ask you, that when the cell division has taken place, or we can say when two daughter cells are formed from one parent cell, whether this daughter cell have all the cell organelles or not? No. When these two daughter cells are formed from one parent cell, all the cell organelles are not present in these two cells. Okay, many membrane coated cell organelles are absent in these two cells. Why? Because all these membrane bound cell organelles, they will disappear into the to the division phase. In which phase of the division that I will discuss to you. But what happens when the cell division takes place? all the membrane bound cell organelles that are present inside the cell, inside the parent cell, they will disintegrate. So they will disintegrate, as a result they are absent in the daughter cells also. But what happens when they are undergoing into G1 phase? All these cell organelles like mitochondria, chloroplast, Golgi bodies, endoplasmic reticulum, lysosomes, vacuoles, all these will be reformed. They will be reformed into the G1 phase. So this is the first thing that you should know, that in the G1 phase all these cell organelles which were disappeared into the division into the cell division phase, they are again formed into the G1 phase of the cell cycle. Clear? So this is the first point that you should know, that all the cell organelles, all these cell organelles which were disappeared, all these cell organelles which were disintegrated, we can say disintegrated, which were disintegrated during division phase, are reformed. So they are reformed into the G1 phase. Clear? Then second thing I want to discuss to you, the size of the cell, the size of the two daughter cells which are formed from parent cell, their size will be same as that of parent cell, or the size of the daughter cell will be smaller than that of parent cell. Obviously, the size of the two daughter cells, they will be smaller than that of parent cell. Means the requirement is that the size of the cell should increase. So obviously the cell size will increase. Okay. But one thing I want to ask you, what about the amount of the DNA? The amount of DNA inside the two daughter cells will be same as that of parent cell, or it will be different? So the answer is, the amount of DNA, or we can say the genetic material, it is same inside the two daughter cells. Okay, not only the two daughter cells, but also same as that of parent cell, we can say. So the amount of DNA will remain same. Remember, the amount of DNA that was present in the parent cell, same amount of DNA is present inside the daughter cell. Clear? But the cell size will increase. So the size of the cell increases. The size of the cell increases. But remember, the cell there will be no change inside the DNA. The amount of DNA will remain the same. There will be no change in the amount of the DNA. That is understood for you. Then, when the size of the cell will increase, obviously that will require the energy. Energy in the form of carbohydrate, lipids and proteins. So what happened in the G1 phase, these three biomolecules, carbohydrate, lipids and proteins, they are formed in the G1 phase as the metabolic activity of the cell is very high. You can see the cell size is increasing. Also they are forming different types of cell organelles. So as the cell is metabolically very active, that's why the formation of carbohydrate, lipids and protein also take place so as to provide the energy for all these phenomena to take place. So the synthesis of carbohydrate, lipid and protein, carbohydrate, lipid and protein take place. So even though the synthesis of different types of cell organelles, carbohydrate, protein, lipid, everything will take place. But remember, there will be no change in the amount of DNA. This is the characteristic event of the G1 phase. There will be no change in the amount of the DNA. Clear? So all these three events will occur inside the G1 phase. So then it will be followed by the S phase. S phase, or it is also called as synthesis phase. By the name itself you can understand that something will be synthesized. That's why the name is given synthesis phase, and that something is very important. That something is our DNA. So in different books, different wordings are used. So in some books it is given synthesis of DNA take place. In some books it is given formation of DNA take place. Even in some books it is given replication of DNA take place. So all these three words you should know. Okay. So DNA synthesis or DNA formation take place. But my question is, how much DNA will be formed? The amount of DNA forms will be double. That also you should know. That in the S phase, the amount of DNA will becomes double. Clear? So that is the characteristic event in the S phase. That DNA synthesis, we can say DNA synthesis, or we can say DNA formation, or we can say DNA replication. DNA replication take place. DNA replication take place. Okay. Then, even I discussed to you that in the some illustration I will give you. That also done. The amount of DNA, the amount of DNA becomes double. So that illustration I will give you, because same illustration in CH10 also. That example I will give you. Means suppose in the G1 phase, if in the G1 phase amount of DNA is 2C, in G1 phase the amount of DNA is 2C, then what is the amount of DNA in S phase? The amount of DNA will be how much? 2C is the amount of DNA in the G1 phase, therefore it will become double, means it will be into 2, means it will be 4C. Clear? But the most important thing is, what about the number of chromosomes? The number of chromosomes also will become, no, the number of chromosomes will remain same. Only the amount of DNA will become double. But here confusion comes, for how it is possible that when the amount of DNA will become double, how the number of chromosomes will remain same. You see, this is a structure of chromosome, but it has not duplicated. It don't have the chromatids. So what happens when the duplication will take place? It will have two chromatids like this. So this is how many chromosomes? One chromosome. This is how many chromosomes? This is also one chromosome. But only thing is that here the amount of DNA has become duplicated. That's why here the chromatids are there, but chromatids are not there. So this is unpaired, means we can say this is unduplicated DNA. This is unduplicated chromosome, and this is the duplicated chromosome. So here you can see the amount of DNA has become double. Also the amount of DNA in this one will be double, since symmetry is there, you can see. But the number of chromosomes are remaining the same. So that you should do. Okay. Now we go to the next point. That I told you the synthesis of the protein take place in the G1 phase. Even I am saying it will occur in the G2 phase also. So synthesis of protein take place in the S phase or not? So usually synthesis of protein does not take place in the S phase, but exceptionally a special type of protein that is called a histone protein. Histone is a basic protein. As with the nature of the DNA is acidic. So as to neutralize the acidity of the DNA, a basic protein that is called a histone, it is produced by the cell inside during the S phase. Even non-histone protein is also formed, but the main credit is given to the histone protein. So that is also the event. So histone protein, histone is a basic protein, remember. It is a basic protein. So histone protein is formed during S phase, What is the role that I discussed to you? Now one more event I will discuss to you, but that is exclusive for the animal cell. If you remember in the last chapter, I discussed about a cell organ that is called a centriole. And the function of the centriole is in the cell division, and that I told you, this centriole is present in the animal cell only. It is present in the animal cell only. So what happened? That the duplication of centriole take place in which phase? In the S phase. But remember, this centriole duplication will occur only in the animal cell. Plant cell don't have the centriole. And where the centriole is present? Inside the cytoplasm. One more thing to tell you, that where this DNA duplication will take place, obviously that DNA duplication will take place inside the nucleus. But remember, the centriole duplication will take place inside the cytoplasm in the animal cell. Okay, so this point is exclusively for the animal cell. In animal cell, centriole duplication, centriole gets duplicated inside the cytoplasm during S phase. But remember, this point is exclusively for the animal cell, because plant cells are devoid of the centriole. So all these are the events that will occur in the S phase. Now next phase of the interphase is the G2 phase. That is the most simplest one. So G2 phase, or it is also called as pre-mitotic phase or gap 2 phase also. Pre-mitotic phase. Why it is called as pre-mitotic phase? Because you can see, now after the S phase, the amount of DNA has become doubled. So what happens when the cell is undergoing for the division, then only it will make its DNA double. So it is of very short duration. This G2 phase is of very short duration. And it is the phase of the interphase that is present immediately before the mitosis. That's why it is called as pre-mitotic phase. And the most important characteristic of the G2 phase is that synthesis of RNA and protein take place. So synthesis of RNA and protein take place during the G2 phase of the cell cycle. So this is the most characteristic event of the G2 phase. Clear? Now some points have been discussed here in the note. That 2 points I want to discuss in the notes. So 2 points you have to write in the notes. First point I will discuss to you regarding some cells that do not complete the cell cycle. You know that in our body some cells are there which are not undergoing cell division frequently. For example, heart cell and nerve cell you can see. So what happened? Whether that cells are living or non-living, obviously that cells are living, then only we are living. Okay. But what happened? They do not exhibit continuous cell cycle. They don't follow the continuous cell cycle. What happened? Especially in the case of heart cell and the nerve cell, when they are formed from the mother cell, 2 daughter cells are formed. Now these 2 daughter cells will undergo in the which phase? G1 phase. So in the G1 phase they will complete all the events. Means they will they will form all the cell organelles. They will increase their size. Okay, they will form protein, carbohydrate, lipid, other things they will do. But what happened? They will suspend themselves from the G1 phase. They will complete the G1 phase. After that, they will suspend themselves in the G1 phase itself. Means we can say they will not undergo into the S phase. And they will undergo into the G0 phase. G0 phase is also called as quiescent phase. It is also called as quiescent phase. So what happened? They will complete G1 phase, but this G1 phase will be followed by the G0 phase. They will be followed by the G0 phase, or it is called as quiescent phase. So in this quiescent phase, the cell will remain as such for rest of their life, and they will not divide until and unless they are called to do so. Means if suppose if there is any injury, then they may undergo for the cell division, or we can say cell cycle. Otherwise they will remain as such in the G0 phase, or we can say in the quiescent stage, and they will complete their life in the G0 phase itself. So remember, such cells are metabolically active. Obviously, if you see the heart cell, they are very much metabolically active because they are doing continuous contraction and relaxation, but they are not completing the cell cycle. So after the G1 phase, they will exit the G1 phase and they will go into the G0 phase or the quiescent stage. So they will not undergo into the cell cycle. So this is the first point of the note. Now second point of the note is regarding how the cell regulates itself between the G1 phase, S phase and G2 phase. Means how the transition takes place between these three phases of the interphase. Means how the cell will control itself that now it is the time for the G1 phase, now it is the time for S phase, and now it is the time for G2 phase. So the transition between these three phases of the interphase is done by a factor, and that is called as CDK. CDK stands for cycling dependent kinase. Cycling dependent kinase. So what happens? This CDK, cycling dependent kinase, controls the transition of the interphase, controls the transition of the interphase from the G1 phase to S phase, and then from S phase to the G2 phase. So all this control, or we can say this, all this regulation of the interphase, not only interphase, this regulation of the cell cycle also, this is controlled by a factor that is called as CDK, cycling dependent kinase. So this is also a point that you should know. Clear? So by this our interphase is complete. Now in the next class I will start from the mitotic phase, and that is nothing but the karyokinesis. Okay, thank you.